On today's Side Talk 2.0, I will be interviewing one of the most prominent neuroscientists, Dr. Joseph LeDoux. We will discuss human consciousness, evolution of human consciousness, inherent biases, and split brain patients. So stay tuned for an informative and exciting interview. Hello, everybody. Welcome to another exciting episode of Side Talks 2.0. My name is Dr. Jack Saxenian, and I'm excited to introduce my guest today, Dr. Joseph LeDoux, who is one of the most prominent neuroscientists studying the brain and human consciousness. He has some fascinating research done on the brain over the last few decades and has published numerous books, including his recent one, which is a very interesting tour of how the human consciousness evolved. So with that and without further ado, I want to introduce Dr. Joseph LeDoux. And if you could please tell a little bit about your background and your research.
Okay. My background is kind of unusual, so I'll start at the beginning. I grew up in a small town in Louisiana and went to college and studied business administration and got a master's in marketing. And in the process of doing that, got interested in like consumer psychology, consumer behavior, and took a psychology class from a professor who happened to be studying the brain and memory. And I kind of fell in love with what he was doing. I had no idea you could study the brain at that point in my life. And so I just threw myself into that and was able to get into a graduate school at the program at SUNY Stony Brook up on Long Island, and moved up there and started working with Michael Gazzaniga, who studied split brain patients. And I know that's a topic you're interested in and asked me to maybe talk about a bit about. So I can just introduce that right now, if it's okay.
Yes, for
Okay.
sure. It's been one of the inspirations personally for me going into neurosciences myself, reading these exciting patient stories. So if you could please.
Sure. So split brain people are people in whom the two hemispheres have been surgically separated by cutting usually the corpus callosum, a fiber tract that allows the left hemisphere to know what's going on in the right and the right hemisphere to know what's going on in the left. Now, in a split brain patient, without the left side typically has language or more language than the right side, and you can hold a conversation with the left hemisphere. But the right hemisphere in most patients is rather non-communicative. It doesn't have a lot of language, certainly not speech. And so it was always kind of difficult to know what was going on in the right hemisphere. But you could do tests like, for example, if you flash a picture of an apple to the left visual field, that means the apple goes to the right hemisphere. Now, in you and me and everybody else whose whose hemispheres are connected, information in the right hemisphere is available to the left by way of the corpus callosum. So an apple goes to your left visual field in your right hemisphere, you can say, oh, I see an apple. But if your brain has been sectioned in this way, and by the way, the reason this is done is to control the flow of seizures from side to side in patients in whom medications and all other efforts to control the seizures have failed. So it's done only in a last-ditch effort to try and bring the seizure activity under control. And it's a pretty severe choice of a person, a patient to make, or the patient's family to make. So whether you want to split your brain in half, that's irreversible. So, but these people were in many cases pretty desperate for some kind of help. And the patients that I tended to work with were usually teenagers that had epilepsy most of their lives and really couldn't lead a life. So this was the hope of an opportunity for a life. Now, I wasn't involved in their clinical work, or their clinical status or anything like that. So I don't really know what the outcome was because after I did those experiments, I went on and did other things. So I don't really know how they did in their life afterwards. But anyway, so the right hemisphere has information. You and I can talk about that information because it goes to the left, and that's where your ability to speak is in most people. But in these people, they say, I didn't see anything. So the right hemisphere has a picture of an apple in its visual cortex, but it can't tell you what it is, but it can reach into a bag and with the left hand and pull the apple out. And there could be an apple and a banana and some other things, or a screwdriver and other things in there, but the left hand will pull out the apple, showing that the right hemisphere perceived the stimulus, but was unable to communicate about it because of speech being in the other side of the brain. So that's the basic finding. Now, those kinds of findings were discovered in the first group of patients in California out at Caltech, where the work was done by Michael Gazzaniga working with Roger Sperry. And Gazzaniga designed all these tests and figured out how to get into what's going on in the left side and what could and couldn't happen when your brain is split.
Let's take a look at some of the patients that Dr. Gazzaniga has studied and watch a video clip in which he demonstrates the different visual perceptions between the left and the right hemisphere and the answers that these patients give to the testing that he does. So let's have a quick look on this clip before we return back to the interview again.
I know the left hemisphere and right hemisphere now are working independent of each other, but you don't notice it. Now, you just kind of adapt to it. You don't have any feeling, it doesn't feel any different than it did before.
7 years ago, Joe had brain surgery to allay the effects of severe epilepsy. His surgeon cut the nerve fibers connecting his left hemisphere with his right. While the operation was a complete success, Joe's unusual case offers an extraordinary insight into the machinery of mind. This fiber system, the corpus callosum, is located midway between the two hemispheres. When it was surgically severed in Joe's brain, the transmission of information between the two hemispheres was halted. Michael Gazzaniga.
What we can do is play tricks by putting information into his disconnected, mute, non-talking right hemisphere and watch it produce behaviors. And out of that, we can really see that there is, in fact, a reason to believe that there's all kinds of complex processes going on outside of his conscious awareness of his left half brain. Joe, I'm going to show you some things. I just want you to tell me what you see. And here we go. You ready? Look right at the dot.
Okay.
Right. Okay, you ready? Look right at the dot.
Grapes.
Good. When Joe focuses on a point, look right at the dot, everything to the right of the point goes to his left brain, the dominant hemisphere for language and speech. Look right at the dot. So we can see here that when we flash a word or a picture,
Tree.
Joe is easily able to name it.
Here we go. See it.
Close your eyes and let your left hand do a little work here. Okay, what do you got there?
Pan.
Okay, very good. Now, when a word or a picture falls to the left of a fixation point, that information goes to his disconnected right half brain. And as we can see here, Joe is unable to name it. Joe is able to draw the picture with his left hand, the left hand getting its major control from the right half brain. What'd you draw?
Okay.
What'd you see?
Wheel on one side. I don't know what I saw on the other.
So even though he can't name it, his left hand is able to draw out a picture of the stimulus of the picture or word that we presented to his right half brain. What did you see?
Saw a hammer.
So just close your eyes and draw with your left hand. Just let it go. That's nice. What's that?
Saw.
Yeah. What'd you see?
Hammer.
What'd you draw that for?
I don't know.
What we have with Joe is just a dramatic example of a neurologic case that really allows you this window into the non-conscious and how powerful non-conscious processes are at influencing our conscious self, our personal self. What Joe and patients like him, and there are many of them, teaches us is that the mind is made up of a constellation of independent, semi-independent agents, and that these agents, these processes can carry on a vast number of activities outside of our conscious awareness. Even though that goes on, there's some final stage or some final system, which I happen to think is in the left hemisphere, that pulls all of this information together into a theory. It has to generate a theory to explain all of these independent elements. And so, and that theory becomes our particular theory of ourself and of the world.
He had moved to Stony Brook, and that's right around the time that I arrived there. And we had a new group of patients that we had the opportunity to study. These patients were operated on at Dartmouth Medical School by a neurosurgeon there named Donald Wilson. And he, he had a group of about, I don't know, I think there were 5 or 6 that we had access to at the time. Some of them were not amenable to being studied, or had other problems that would impair the ability for them to participate in these experiments. So there were 2 or 3 that we focused on at the beginning there. Now, what we decided to do in these group of patients, it's not like we really went into it with this idea, but the idea that came out when we started studying these patients, well, in the California patients, the idea had been to see what the consequences were of taking, splitting the brain in half. But in these patients, we decided as the experiments rolled on and became, when results started coming in, we said, well, we have the opportunity here to ask a different question, which is, what are the implications for the normal brain when this happens? And so Gazzaniga and I wrote a book at the end of all of this called The Integrated Mind, where the idea was to try to use all this information for understanding what goes on in our minds. So here's an example of why we went down that particular road and what we were thinking about. It had to do with one patient who had the ability to read in his right hemisphere, but in his left hemisphere, he could read, he could write, he could understand language and so forth. But in the right hemisphere, he could only comprehend language. So he could understand and he could read. In other words, he could comprehend spoken language and read written language, but he couldn't speak with his right hemisphere. Only the left could, as in the other patients. That was the opportunity to put more information into the right hemisphere, because we could put questions into the right hemisphere, like, who are you? And then we gave the boy, the teenage boy, a set of Scrabble letters where he could select his answer. So we asked him, who are you? And the left hand reaches out and pulls out the letter P-A-U-L. So he spelled his name, Paul. So that meant that there was an entity, a mental entity over here who knew who he was. He had a sense of self because he knew his name. He knew what we were talking about when we said, who are you? It's actually a complicated question if you think about it. You have to understand, of course, all the syntax of the sentence, but you have to know what that means. Who am I? And, without having a kind of history that right hemisphere had gone through to have a sense of self over there, he couldn't have answered that question. Of course, we knew the left hemisphere had a sense of self because we talked to him all the time, but the right hemisphere, we had no idea. And then we asked the left and the right hemisphere separately, what do you want to be when you grow up? So, we just asked the left hemisphere verbally, what do you want to be when you grow up, Paul? And he said, well, I want to be a race car driver. But with the language presented to the left visual field and the Scrabble letters, we had the right hemisphere respond to the question. And he said, the right hemisphere said he wanted to be I'm sorry, the right hemisphere wanted to be the race car driver. The left hemisphere wanted to be a draftsman, an architect.
Wow.
So that, you know, this isn't consistent with the old left-right idea. The left is verbal and the right is spatial, because it was the left hemisphere that wanted to be the architect. So let's just not go down that road. But the here we not only had a sense of self in both hemispheres, but also a life ambition, a goal for the future. And those are two kind of hallmarks of human consciousness.
So I have a couple of follow-up questions.
Yeah, okay.
One is that, you know, people, like you mentioned towards the end, that, you know, we tend to think of the right being more spatial, and you know, people think of lateralization as, you know, mathematics on one side and creativity on the other side. So what you're saying is that there is lateralization, but it's not the way we think about it, right? In terms of the brain.
Yeah, I think it kind of got overblown in the 60s, and we never recovered from that. You know, I have all these books written about, you know, drawing on the right side of the brain and all these things, personality differences. You know, it's not like each hemisphere is a different, specialized for a different personality. There are differences. I mean, you know, I haven't worked in this area since 1978. Sure. So I don't know how current my knowledge is, but at the time, this is what we thought. That there are differences between the hemispheres, but they're more about what are the specialized capacities, like, you know, the ability to speak and usually to process language in the left. This kid had language and comprehension in the right. Now, the answer to why that occurs is that early brain damage causes a shift in functions. So if you have damage in your left hemisphere and are missing something there, it can cause the right hemisphere to have to acquire that capacity. And you know, I don't know exactly why this kid developed. I mean, he had good language in his left hemisphere, but that's the only explanation I can come up with. There was the epilepsy early in his life somehow triggered a reorganization of the brain such that he was able to to comprehend language on the right side of his brain. But anyway, so the idea is that, you know, you got language, usually you have language on the left and spatial stuff on the right, but it's not like the entire left hemisphere and entire right hemisphere are focused on those things. I think it's just kind of like an isolated couple of capacities.
So what does this mean in terms of, I know this might be a little bit of a deep question, in terms of human consciousness? Does that mean the two sides are separate entities of consciousness that are
No. I think, you know, when the brains are connected, the two hemispheres are connected, it's just one brain. Mm-hmm. But what you're doing is kind of artificially forcing the person to have two separate mental spheres because they can't communicate, at least not internally. But they can use external behavior to communicate. And that's the next experiment I'd like to tell you about, where we put two stimuli, one in the left hemisphere and one in the left visual field and one in the right visual field, and showed them to the patient simultaneously. So in the right visual field, going to the left hemisphere, was a chicken claw. Like a claw. In the in the right visual field was a chicken, picture of a chicken head. In the left visual field, going to the right hemisphere, was a shovel. Sorry, a snow scene. Okay, so we had a chicken and a snow scene. That's what I mean. Sorry, it's a long time ago. So the two hemispheres each saw a different stimulus. Now, the two hands reach out and point to different things. The right hand connected to the left hemisphere saw the chicken, pointed to a chicken claw. Mm-hmm. The left hand connected to the right hemisphere pointed to a shovel because the right hemisphere saw a snow scene. So the guy's looking at his hands and he says, why did you do that? And the left hemisphere said, well, I saw a chicken, so I picked a chicken claw. And you need a shovel to clean out the chicken shed. So he was a country boy. He knew that, you know, chickens are a messy animal and they live together in this chicken coop and you got to clean that out somehow. And so what happened was he observed the left hemisphere that you're talking to, observed that the left hand, which controlled by the right hemisphere, had picked a shovel, and he incorporated that behavioral response into his mental model in the left hemisphere about why he would pick a shovel. Well, I just saw a chicken and a chicken claw, so I picked the shovel. So that's when the light went off in our minds, and we said, okay, we're not studying split brain patients here. We're studying like the essence of human consciousness, how we go through life. Because what we just saw was the conscious mind narrating a story about one's behavior to make it make sense. So our idea was that that's what happens to us all the time. We don't have conscious access to all of the systems in our brain that are generating behavior. And so we think we have free will, right? So we have to, consciousness is very protective of free will. That's one of its prized possessions. So to keep that sense of mental unity in the face of mental diversity, where the conscious mind doesn't have access to everything, one of the things consciousness has evolved to do is to keep us unified by making sense of all things we do. And we thought, sorry, what was your question?
I said making up stories to fit.
Yeah, making up stories. The narrations, these are stories. We tell our life story as we go through life. We learn our life story and we tell it. And we thought, well, maybe one of the systems that would be doing all of this, that would require these narrations, would be emotion systems that are generating behavior unconsciously and then causing, you said, well, why did I do that? We often don't know why we did something, but we know we did it. And so we generate a story. There have been tons and tons of studies since the 1950s and 60s on this topic, basically falling under the rubric in psychology of cognitive dissonance. This is a theory that Leon Festinger proposed in the late 1950s, which says that when you have two mental events, or you have a behavioral response and a mental belief that are contradictory, that don't go together, you have to resolve that because it's a source of dissonance. And so the way you resolve dissonance is to discount one or the other. And so in the case, or to do something with it to make it all make sense. So in the case of the split brain boy, what he did was he did something to resolve that dissonant state that would come from his body producing a response that he didn't generate. So that's a source of uncomfortableness if you believe in free will in yourself, right? I didn't do that. Why did I do that? So, but without blinking an eye or even, thinking about it, he just said, I picked the shovel because you need to clean out the chicken shed.
So not to get political or social, but does that sort of explain some level of like inherent bias that people have it, but don't know that, they have bias towards certain things or whatever. And their brain kind of explains it, but where it's coming from, they don't really know it because it's sort of hidden.
Yeah. So we have all these things in us that we don't know or understand very well. So if you have a racial bias, for example, but it's not tolerated in your society, then you explain that as, oh, that's not how I am. That just happened or something like that. Unfortunately, this kind of thing is tolerated in our society now, so people don't have to explain it anymore.
Sure.
Things are going. So anyway, yeah. So that's what led me to begin studying emotion. It's like we said, okay, we were at the bar that night after we'd go to the bar after we test the patients to kind of think about it and talk about it. And at the bar, I think Mike said, we were talking about this, emotions are interesting and maybe this is an emotional response. And Mike said, there's not much research on emotion in neuroscience these days. And that's when the light bulb went off in my head. I said, that's what I want to do. And there weren't good techniques for studying the human brain. There was no brain imaging or anything in the late 1970s. Or maybe there was, but it's very limited use. I think CAT scans were available, but that was not for function. So I decided that I would, after I graduated, I was going to turn to animal studies and pursue how emotions work in the brain. So I went to Cornell Medical School, and I worked in a laboratory of neurobiology there. And I spent 12 years, which was good because this had every technique known to neuroscience. And since I had no real, biological science training and very little psychology training either because of my background in business and so forth, I spent that 12 years learning neuroanatomy, neurophysiology, how to test behavior in animals and all those things. And I was able to do it because the lab was well funded. I was able to get some of my own funding, and I was just able to kind of spend 12 years slowly building my credentials and my expertise. And so everything I learned was learned because it was relevant to what I was doing in the moment, not because it was book knowledge. So I didn't have any book knowledge about the brain. It was all
Which
on
is,
the job
I think,
training.
somewhat good, right? Because you didn't go in with any kind of bias. You went in.
I think it's a perfect way to do it. I don't think it would be possible these days because there's so much students have to know to get into graduate school, math and molecular biology and all these things. It would never happen today. So I started doing all this, and the research, I decided to follow the stimulus. So the research I decided to do was to take a stimulus like a tone, pair it with a mild electric shock, and you only had to do that one time, and a rat would start freezing to the tone. His blood pressure and heart rate would rise in the presence of the tone. So this so-called fear conditioning task was a very good way to take a stimulus that had no meaning and quickly give it a meaning for the rat. And then the rat would have that memory really the rest of its life. I mean, it's kind of a lifelong thing, learning about danger. It's not very adaptive to lose that kind of information. You got to keep it stored, Now it can weaken if you present the tone over and over again. The rat will stop responding, but you can always bring it back by putting the rat in the context where it occurred, or other things that would revive the memory. So the memory is always in there. And so what I decided to do was to, when you work on split brain patients, you got stimulus coming in here, going over here and crossing. And right. So it's all about tracing the flow of information. So that's what I wanted to do in the rat. I started at the ear, presented a tone, started at the ear, and tried to follow where in the auditory system that tone went to before it branched out of the auditory system. And so rather than starting at the ear, I started at the auditory cortex, the highest level. Lesion the auditory cortex had no effect on the rat's ability to learn to respond this way. That was a surprise because in that day and age, it was thought that you had to get the tone all the way to the auditory cortex before it could do anything meaningful, because that's where meaning was added. When we lesioned the auditory thalamus, however, that blocked the ability to learn. And every structure below that also blocked it. So the sound had to go into the auditory system to the thalamus, which is the last station before the auditory cortex, the medial geniculate body, as it's called, the auditory thalamus. And but it didn't go to the auditory cortex, so it didn't have to go to the auditory cortex. So where did it go? Well, what we had available at that time was something brand new, which was something called axonal transport. For studying connections in the brain. So you could inject a chemical into the auditory thalamus in this case, and it would be picked up by the neurons there and shipped down the axons to the end of where the axon ends, where the nerve fiber makes a synapse with another cell. So we injected the auditory thalamus, and of course, we found a lot of these fiber connections in the auditory cortex, because that's the main output of the auditory thalamus, but we also found them in the amygdala. Wow. And so we thought, oh, okay, so maybe that's how the tone is, that's where the tone is going after the auditory cortex, after the auditory thalamus. And sure enough, we lesioned the amygdala and the animal couldn't be conditioned. The tracing studies showed that it was not in the entire amygdala, only in one small part called the lateral nucleus of the amygdala. When we lesioned that little input region, we also disrupted conditioning. Then we traced the connections from there to other parts of the amygdala and found the output connection, which was the central nucleus of the amygdala. And it connected with the midbrain periaqueductal gray area to control the freezing response. It connected with the lateral hypothalamus, and from there to the brainstem to control the autonomic nervous system responses. Yeah.
If I may cut you off and ask you a really quick question in regards to this. So
that's fine.
In this context, you studied a stimulus which was very unpleasant to the rat, right? So
right.
You would see the signal going to the amygdala because it's presenting some kind of danger. But does that mean every kind of sound would have to go through all of these, or?
Yeah, good question. Yeah, very good question. So the idea is that, let me add another piece to the puzzle. So what we found was that while the auditory cortex wasn't required, it was involved. So if we lesion the part of the auditory thalamus that projected to the amygdala, the animal could still use the information going to the auditory cortex and to the amygdala. So I gave a name to these two pathways. One was the low road. That was the pathway from the thalamus to the amygdala. And then the pathway from the cortex, auditory cortex, to the amygdala was the high road. The low road was a quick and dirty pathway. It allowed a stimulus to get there within a few milliseconds, whereas to go through the auditory cortex, it takes a little longer, but it provides a much more elaborate picture. That's why both in life are really needed. So, you're walking through the woods and you are about to step on a snake, but through the visual system connections to the thalamus and to the amygdala, you freeze right before you step on the snake. And then you realize there's a snake through the more elaborate cortical pathways. And if it turns out it's a stick around, a curved stick instead of a snake, then you just keep walking on. But it's these two pathways that give you this kind of information about what is there. And the idea is that what conditioning does is it takes certain sounds, and this is the Pavlovian conditioning that we use, it allows features of the particular sound that is paired with the shock to get into the amygdala. So the amygdala is, if you put electrodes in the amygdala of a rat or any mammal, what you find is that the cells are really, really silent. There's almost no spontaneous activity. It's very hard to elicit a response from the amygdala. And the reason for that is there's a tremendous amount of the neurotransmitter GABA and GABAergic cells in the amygdala to keep it clamped down. So everything that comes through doesn't get in because it's inhibited. It's a gate that prevents it from entering the amygdala. So to open that gate, you need something like conditioning to allow that stimulus to pass through to open the gate. Most stimuli don't. Only those that are associated with danger have the gate opening. So does that answer your question?
I guess that's a great thing, because otherwise, if the amygdala was sort
No,
of
it would always be
active, you would never be able to move or do anything in life.
Yeah. Okay. Yeah. So we found three different outputs of the amygdala, of the central amygdala. One to control the autonomic nervous system. Actually, we found a sympathetic and a parasympathetic output. We found a behavioral output for freezing, and we found an output to control stress hormones from the pituitary gland. So these are three separate outputs. When I say we, I mean, I did a lot of this work, but there were other people in the field as well that were finding all these things. So it's not just me. So we were able to, within a very short number of years, move from the ear all the way to the motor systems that control the responses, with the amygdala in the center of all this. Now, the amygdala was involved regardless of what kind of stimulus you used and what kind of response you measured in these tests. So it was a stimulus and response independent part of the pathway. And that's why we thought it was such a crucial hub of this whole thing. Now, over the years, the amygdala, and my work contributed to this, came to be known as the, quote, fear center. And I think, that's something that really bothers me, because I feel I contributed to this cultural meme but it's not something that I endorse today. And even back in the early days, I wasn't quite endorsing it in the way that it is known. So what I was always talking about was that the amygdala was one of these unconscious, non-conscious circuits that would detect and respond to danger. And when a response is generated, it would be something that consciousness would have to interpret, as in the split brain patient. So I described it as an implicit fear center. You know, the circuit was unconsciously processing the danger and producing the responses. But that was too awkward or too complicated. People said, it's a fear center. Forget the implicit. That's too complicated. And so that's what the amygdala became, is like a fear center.
I mean, it's
and,
still taught that way in medical school and graduate school, you know, and it's unfortunate.
And most of my colleagues, you know, I've been on this crusade since 2012, because after, you know, I started doing this work in 1980, early 80s, and it was always, you know, bugging me that people were like saying, oh, let me introduce you to Dr. Joseph LeDoux, who's discovered how our feelings of fear come from the amygdala. No, that's not what I... So it was always bugging me. And so finally in 2000...
I must admit, there's nothing fearful about you, so...
Okay, good. So in 2012, I wrote a paper called Rethinking the Emotional Brain, in which I said the amygdala is not a fear center in the sense of a conscious fear center. It's detecting and responding to danger. And what we should do is we should call it a defensive survival circuit. It's a circuit that detects the threat and controls the responses to keep you alive. It doesn't do this to make you feel afraid. And that's, you know, and I've been writing about that over the years, but ultimately, after I wrote a book called Anxious in 2015, I started working on this new book, which we're talking about today, The Deep History of Ourselves, the 4 billion year story of how we got conscious brains. I started working on that because of this idea of survival circuits. I said, well, how far back in evolution does the survival circuit go? So we had known that, for example, work that Eric Kandel at Columbia University has done got awarded the Nobel Prize for this work, and the marine snail or slug, the Aplysia californica, on Pavlovian conditioning and other forms of very simple learning in that invertebrate animal. The molecules that they discovered in the Aplysia that are involved in this Pavlovian conditioning were clues that we could now test in a mammal. It would be too hard to figure it all out in a mammal, and that's why Kandel turned to the Aplysia, because it would be a much simpler thing, and he made great progress. So we took advantage of that and said, okay, well, he's got things like MAP kinase and protein kinase A and all these enzymes that are important for learning and memory in the Aplysia, CAM kinase, all these things. So we said, let's try them in the rat amygdala. And sure enough, they were all involved. So you start thinking, well, what is, you got two different lines of animals. Mammals are vertebrates. Aplysia and flies and all these other animals are invertebrates. These little creatures that aren't vertebrates. They don't have a backbone. They don't have a nervous system the way ours, a nervous system, a spinal cord, sorry, and inside their backbone. But they have a nervous system. But where does that, how do we get the same molecules in these two very distinct lines? It turns out you got to go back about 630 million years ago to find a common ancestor that gave rise to those two lines. And that common ancestor is a flatworm, which is it's believed to be the first bilateral organism. So the bilateral organism has a left, right, front, back, and top, bottom. So like us, top, bottom, left, right, front, back. And so the bilateral organisms, the earliest bilateral organisms, according to genetic evidence, and that's one of the beauties of all this, fancy genetic stuff people can do now. You can trace the genes that are present in other organisms. And so Seth Grant, who had worked with Eric Kandel and at some point moved to the University of Cambridge, was a good friend. And I was at Cambridge, and that's when we really became friends. And we talked a lot about all this. And I learned from him about this early ancestor that was the common ancestor for these groups that had some of these same molecules. Now, what Seth was interested in were the molecules underlying what's called the Nmda receptor, N-methyl D-aspartate. That's the glutamate receptor that's involved in synaptic plasticity in our brains and also in invertebrates like the eplesian flies and so forth. So Seth was tracing the origin of this Nmda receptor, and sure enough, it was in these early bilateral animals, but also in what's called radial animals, like jellyfish. They don't have a left and a right and a front and back. All they have is a top and a bottom. A jellyfish is circular. So, and it has, some of them have eyes all around their head. So they're not oriented the way we do. But they have a nervous system. They can learn about danger and escape from it and do all that. And they have components of this Nmda receptor. Now, before jellyfish, we get to the first group of animals, which is sponges. Now, sponges don't look like much, but they are animals. And they don't have a nervous system, though. And it's unclear whether they had one long ago and lost it. Sometimes that happens. Features get lost with change. Or if I think the preferred theory right now is that they had that they did not have a nervous system, but they did have all of these NMDA components and other synaptic plasticity components, even though they had no nervous system. And consistent with that is that the ancestor of the sponge is a single cell protozoan called the chenoflagellate. Now, this guy has no nervous system. It's one cell, so you can't have a system if you're only one cell. Or you can have this, like components of that cell. And it has the Nmda receptors, or components of the Nmda receptor, and it learns. So without a nervous system, you can learn and survive. You can detect danger and do all kinds of things. You can detect.
Can you run for a presidency?
You probably can. In the protozoan world. Maybe in the sponge world, they've got a protozoa running for president. Oh, that's not good. All right. Anyway, so the point is that we're at a point where there's an ancestor that's not an animal that is capable of learning and has some of these molecules that are used by animals to learn. So this is like, what evolution does. It takes stuff that's already there and uses it for a new reason. So nervous systems were particularly useful because it allowed bodies to get bigger. If you can only communicate by chemicals, it's a very slow process. So plants or another kind of multicellular organism. There are only three kinds of multicellular organisms: plants, fungi, and animals. Fungi are actually closer to us biologically, evolutionarily, but they get stuck with the plants in the grocery store. So it's kind of
right.
So the interesting thing. Well, so we're talking about trees now. So a plant can move with the sun, but it's a very slow process. It requires chemical, allows chemical diffusion throughout the plant body, and changes in this part of the body have to be slowly, picking up the sun with the leaves and so forth. And it's got to, slowly send chemicals down the stem to move the stem slowly with the sun. It's a very slow process. An animal can respond like that. Why? Because a stimulus on any part of our body can go to the brain and cause a response instantaneously. And that's the beauty of a nervous system. It allows rapid communication. It's based on the same thing as in a sponge, ultimately, chemical communication, but it uses the electrical signal from the cell body to the end of the nerve terminal to rapidly cross distance. And so you can get information one spot, then to the next spot, very quickly, but you still have that chemical link at the end The chemical is always important, not always, because there is electrical communication between cells. But for most kinds of transmission, chemical communication allows you to cross distance very quickly and allows you to have a very big body that can do things very quickly. We couldn't have that if we were, it would be a tree if we didn't have a nervous system.
So in essence, evolution took that single cell organism and kind of copy pasted to generate a nervous system, which allows for bigger bodies to be able to respond.
There were steps. So what happens is if you think about, for example, a sponge and a jellyfish.
Mhm.
The adult forms look nothing like each other, right? They're very different. But in the larval phase, they're similar. So they're both small creatures that are free floating in the water. They swim. And they have this kind of almost identical shape. And the idea is that the way you got from, a sponge to a jellyfish is by developmental modification of the larva. It was a small step from changing the sponge larva to a jellyfish larva. And then a genetic program with epigenetic regulation allows the body to be different, to satisfy the needs relative to the niche in which the organism is developing. Now, the same thing happens because the protozoa that is the ancestor of the sponge looks identical to a cell component of the sponge.
Wow.
And so, again, it has to do with the protozoan ancestor is the ancestor of a sponge larva.
Wow.
That then has a developmental program that makes the sponge. So that it's a set of connections. So now we're about, I don't know, a little, 1.5 to 2 billion years back from where we started. But we've got these protozoa. Protozoa are eukaryotic cells. That means they have a nucleus and a cytoplasm with organelles in it, like mitochondria and stuff. And that is true of us as well. So all animals and some protozoa, so all animals and protozoa are eukaryotic cells. And eukaryotic cells are important because they invented sex. They invented sexual reproduction. And that became increasingly important when we start to get to the arrival of animals, because you are in general for multicellular organisms, because you have to take that single cell, let's say the protozoan, is gonna, let's start. Most single cell organisms like bacteria reproduce by dividing.
Mm-hmm.
Cell divides in half, and then you have more bacteria. They just keep dividing. But so in a sense, the first bacterial cell is the mother of every bacterial cell, because that first one divided, and then the two daughters divided, and they kept dividing. So that there's that lineage from the first bacterial cell that goes all the way to every bacterial cell alive today. But for eukaryotic cells, every cell has a different, every new cell has a different mother, unless it's it could be a sister. You could have a brother and sister in that way. But there are every generation has a new mother. So that's one way that the bacterial cells differ from these eukaryotic cells like protozoa. But another way is that that allowed the single cell with its genome to now be fertilized by another cell to create
brand new
an egg. And once you have an egg, you have the genetic program for a whole organism. If you're a protozoa, it only makes another protozoa. But it's a really complicated set of events, but you go from unicellular organisms to cells, to organisms with multiple cells. And it was slow. There were just a few cells and others that had many more. But you've got to start with that fertilized egg so that the organism can come from that one cell. You've got to have the DNA in there that's going to give the developmental program. Let's say at first it gave the developmental program for 2 cells or 3 cells. And then from there you could get more and more, and then that's how it happened. So that happened in animals, plants, and fungi, multicellular organisms that are macroscopic. Everything else is microscopic, bacteria and protozoa and so forth. Now, okay, a protozoan cell has to do 5 things to get through the day: detect danger, incorporate nutrients, balance fluids and ions, thermoregulate, and reproduce. So obviously you have to detect danger, and they do that by moving away from it, and they can learn about what's dangerous and move away from it. They incorporate nutrients by, if they're swimming along in the environment and they hit something toxic, they swim away. If they hit something that's nutritional, and by how they know that, because it's programmed in their cell membrane, it's that one cell, so it's just detecting what's useful and dangerous. And if it's useful, it just keeps moving towards the concentration and, gets some food, energy source, and goes on about its business. It has to balance fluids because the cell, if it has too much salt or too much water, it will either explode or collapse. And so you got to have the right balance of salt and water inside the cell. Thermoregulation is required to keep the body temperature of the cell in roughly the right range, and they do this by moving around and to find the right temperature so that they can have their enzymes working properly to keep the cell going. And finally, they have to reproduce for their kind to survive. Now, when we do these things, we talk about fear, hunger, thirst, whatever, warmth and cold. If warmth feels good, cold feels bad.
Hypothalamus almost.
Yeah, and sexual pleasure, But when we do that, we're projecting our mental states backwards on evolution. And we'll come back to that, but I just wanted to plant that seed because now we go back to bacteria, which are the ancestors of these eukaryotic cells. So 2 kinds of bacteria existed 3 billion years ago, 4 billion, about 3 to 3.5 billion years ago. One was called bacteria, and the other was called archaebacteria. And what happened is about 2 billion years ago, an archaebacteria and a regular bacteria came together. Either the bacteria was a parasite and it went inside the cell to survive on that cell, or the cell, the archaeal cell, absorbed it, so basically swallowed it and took it in. We don't know which way it went. But in either case, inside that cell, now you had a bacterial cell inside an archaeal cell. Now what happens is at the same time, the membrane began to unfold and surround the DNA, because in bacterial cells and archaeal cells, the DNA is free floating in the cytoplasm. So you have to, in order to create a eukaryotic cell, the DNA has to be encompassed, encircled by a membrane. And so the cell membrane, comes in and starts surrounding that. And that's what eukaryotic cells are known for, having a nucleus. That's what eukaryote means, is like, first kernel, or, a kernel. And so but the most important thing was, I think, that the bacterial cell that went in there became the mitochondria of the eukaryotic cell, the energy machines. Mm-hmm. And in order for that to happen, the 2 cells had to each shed some genes. They had to lose genes, because in order for a cell to be compatible, to survive, its components have to be compatible. Mm-hmm. So you had to shed enough genes for the 2 cells to be compatible. And that's one of the things that is true of a multicellular organism. Every cell in the body is compatible because they have all the same genome in them. And without that, you couldn't survive, because your cells would all be in conflict. The cells have to get up, the eukaryotic cells have to give up, if for a multicellular organism, the eukaryotic cells have to give up their reproductive rights and give those over to one cell that is going to be the sperm or the egg in a sexual organism. And all the other cells now have sacrificed their reproductive rights and for the good of the organism. They each have a job, but it's not reproduction. Some are doing feeding, some are doing locomotion, but they all have the same genome, and that's why a body works.
Unless you get cancer, right?
Yeah, that's right. Cancer is an example of a rogue cell coming in and destroying the ecology of the whole thing, changing the physiology of the body. Now, so I'm just going to throw this other thought. I don't know how far we're going to get on all this because it's taking a while, but the human brain and its conscious mind is the only entity in the history of life that has ever been able to choose to end its life. In other words, we have this rogue component. So one part of the body can choose to end the work of the rest of the body. And to do that means to go against all of the evolution that built a body. There's never been another animal that could willfully and intentionally choose to end the life of the body. There are animals that go through these behaviors that look like they're suicide, but it's been pretty convincingly argued that you can't prove the intent, that there was an actual intent to do it. They do things that end up killing them, but they didn't have the intent to go into that situation to terminate their life. So, and that is what our conscious mind can do. It can do things that that no other animal can, such as end your body, but it can also decide to build a building. We can envision the future. We can do all kinds of wonderful things. But our conscious minds are also the source of all of our worst tendencies as well. Egotistic behavior, narcissistic behavior, selfish behavior. So our conscious minds have created a world for us that is quite phenomenal in terms of all the amazing things that we have available to fight off infections, to live in peace and harmony without having to deal with predators every time we go out for food, to live amongst each other, but also to fight with each other, to disagree with each other, to kill each other for political or religious or any other thing that is on the mind. So our minds are incredibly powerful. And right now we're choosing to destroy the planet that our ancestors, our descendants will inherit from us because we have short-lived goals. We're not taking the big picture. Now, the only way we can stop this and save something for our descendants is it means every human group has to come together. We have to put our conscious minds have to decide that this is what we want as a species, is to move forward in a more productive way. I don't know how we're ever going to do that. And it may mean that, like many organisms, they come and go. In our life cycle, I think we've already kind of exceeded the ideal lifespan of a species. We may just, we may not be here much longer if we keep up like this.
So based on your book and what you've just explained, does that mean human consciousness can be traced all the way back? I know this is a major sort of conclusion coming from, at least from my end. Does that mean it goes all the way back to billions of years, or just basic functions go back?
Right. So let's do what I did in the book, which is I got to the point where, excuse me, I got to the point where I had decided that we have to go back to the beginning of life, to the first bacterial cell that gave rise to the entire history of life. And at that point, I then had to climb back up the tree of life and see how behavior changed as we went from bacteria to bacteria and archaeal cells together, from those to protozoa, from protozoa to sponges to jellyfish to bilateral organisms to invertebrates like flies and bugs and so forth, and vertebrates like us. And so I climbed all the way back up. And what I argue is that, things like emotion and consciousness, when we are talking about them as a human, are only present in humans. Only humans have human consciousness. Only humans have human emotions. The problem is, if you want to claim that other animals have those, there's no way to do it.
I mean, I don't even know if you're conscious, right? I mean, if we're really being
Right.
specific, I only know that I'm conscious.
That's right. And that's an argument that a number of people have made to me. But there's a difference. You and I are humans, and so we have a human brain. But a rat and a person have different brains. Every species has its differences with every other species. That's why it's a species. So, you know, we have no problem recognizing the difference between a chimpanzee and a human.
Right.
And yet, you know, people are wanting to project our mental states onto chimpanzees. I'm not saying we shouldn't, but I'm trying to point out that we're overvaluing our mental states when we do that. That Darwin, I think Darwin really did us a disservice. And of course, he was a great biologist, but it's been said he wasn't such a good psychologist. So he can't be good at everything, right? So
Right.
he said that emotions are states of mind we've inherited from our animal ancestors. And that statement has been, you know, the basis for the entire history of emotion research, that we inherited these mental states, these emotions from other animals. But really all we know about another animal is from its behavior.
Right.
You know, you see a dog on the street, has been hit by a car, he's writhing, he's in pain, he's suffering. You feel so bad for him. But what are you seeing? You're not seeing his pain, you're seeing his reflexes. His reflexes are causing all those body movements. I'm not saying he's not in pain, but you don't see that. All you see is outward behavior. Now, you know, in my research on the amygdala, to go back to that, we know that in a human or an animal, when you lesion the amygdala, if the amygdala is damaged in a human or in an animal, that the organism will no longer respond to a threat in the same way. So a rat will no longer respond to a tone paired with a shock. A human will no longer respond to the tone paired with a shock either.
There's people that are born without an amygdala, right? Cliver Bucey.
There's a condition, it's a dermatological condition that causes calcification. And for reasons that I don't think anyone knows why, the amygdala becomes calcified in these people. Now, one of those people is a very interesting case because for many years, studies of her suggested that a person without an amygdala couldn't feel fear. But then there was another study that was done that showed that if you bypass the amygdala and induce a kind of state of fear chemically, like you would do to induce a panic attack in someone, to simulate the gases and so forth, the blood gas concentrations that cause panic attacks. If you simulate that in this woman, she still has a sense of fear and panic. And my colleague Liz Phelps, a number of years ago, studied a bunch of amygdala damaged people as well. They could also report feeling fear. So, and I want to give you some other evidence so that if you take stimuli and use what's called subliminal presentation, and a typical psychological experiment, that would be a very brief flash, like 20 or 30 milliseconds. You know, that's 20 parts of 1,000 in a second. So it'd be a millisecond, so 1,000 parts, so 20 of those little parts. So it's very fast. So the person under those conditions isn't able to report what was presented and doesn't feel fear. But if it was a picture of a snake or some other threat, the amygdala is activated and the responses are expressed. So the amygdala, fear is not required for the amygdala to do all of that. Fear is not in the amygdala making you do what you do. Fear is your understanding, your conscious awareness that you are in harm's way. So when we project our fear onto other animals, or our love or empathy and all these other things, we're relying on behavior. But the amygdala example shows that that's not a very good thing, because what it's showing is that the conscious experience and the behavior are produced in parallel, not because they're in the same circuit, but because they have the same starting point. A stimulus in the world goes to two different systems. One is producing the feeling by the cognitive interpretation of what's going on, and the other is producing the responses by going to the amygdala.
And so really last kind of quick question, how are mirror neurons sort of fitting into this story, or do you don't?
Mirror neurons are a very interesting scientific area of research that shows that, people and monkeys have neurons that mimic the motor activities of another organism. So these cells will fire when a monkey makes a movement, as if it's recording that in your brain to make that movement. So the greatest success in that area was with these kind of motor mimicry things, but it quickly became fashionable to talk about it as, the source of empathy and all kinds of other things. But I think some of that has been dialed back in terms of what it all means. So, scientists and lay people alike are very prone to running with an idea. And, we all inherit a kind of knowledge, or what's called theory of mind, and an understanding of minds from our culture. It's called folk psychology. We know we've got all these, common stories about what we do and why we do them. We run from the bear because we're afraid, and so on and so forth. But if all we needed was folk psychology and intuition to understand the brain, we wouldn't need science. Right. So we have to go in and analyze what's going on. And what I'm trying to say is that we can't simply depend on behavior to tell us that a mental state that's happening at the same time is the cause of that behavior. Because scientists are taught from day one, distinguish correlation from causation. And what we're doing is looking at the fact that usually when we're running from a bear, we're afraid. So, but because we have this folk psychology that tells us fear causes us to run from the bear, we overvalue instances when those two occur together, and we discount the ones where they don't go together. It's a kind of confirmation bias. We expect fear to be the source of our behavior. If it's not, we say, oh, well, I must not have been afraid, whatever. You just dismiss it. It's one of those kind of split brain narrations that we generate to kind of make it all make sense.
So really quickly, can you tell people where they could find this fascinating book, where they could buy it? And I know you do music, and I've listened to a bunch of them. It's actually really fascinating to see the words and how they tell the story about the brain in a very creative way. So can you tell us a little bit about that before we end?
Oh, sure. Well, in 2006, another professor at NYU and I were getting together and playing for Christmas parties and stuff. And I got invited to give a lecture in Brooklyn at something called the Secret Science Club. And they said, we'll have some entertainment after. I'll bring the entertainment, I said. Okay. So Tyler and I, Tyler Volk, a professor of biology, and I, started working together on this. And Daniela Schiller, a postdoc, arrived around that time, and she was from Israeli, and she was a drummer. She was from Israel, and she was a drummer. So she joined us, and she had a research assistant that was a bass player. So she joined us. So that was the birth of the Amygdaloid. So we went to Brooklyn. We played this set. We had this idea we were going to play heavy mental, which is
I like that.
Music about mind and brain and mental disorders. So we played some classic rock songs like Manic Depression, 19th Nervous Breakdown, Mother's Little Helper, things like that. And I wrote a couple of songs. One was called The Mind Body Problem, and the other was called All in the Nut. So these were All in the Nut was about the amygdala and fear. That was my, bad judgment on my part. We don't play that anymore because it gives the wrong message about fear. And the other song was about the mind-body problems. But all of these are kind of love songs. So the mind-body problem is, my body wants you so, but my mind says no. The woman had left him and she wants to come back. Anyway, so these are all love songs about mind and brain and mental disorders. And we have a bunch of albums. I don't know, I think I've released 7 albums, either individually or with the Amygdaloids. All of these can be found on Joseph-LeDoux.com. This Joseph-LeDoux.com. There's neuroscience, there's books, and there's music. And if you want to go straight to the book, it's deep-history-of-ourselves.com. But you can find that on the Joseph Dash.
I'll put all the links on the
Okay.
bottom for people to.
And of course, you want to buy it, you can. Oops. And of course, if you want to buy it, you can just go to Amazon, and there it is. Or Barnes & Noble's your favorite distributor.
Awesome. This has been really, really wonderful. And I know I could ask you 1,000 more questions. We could go on the whole day.
It's been a lot of fun. Thank you for having me.
Thank you so much.
Good luck with your work and your podcast.
Thank you so much. Thank you so much.
Thank you. Bye.
Have a wonderful day, Dr. LeDoux.
Thank you. Bye.